The hematoxylin and eosin stain is a routine stain performed in all histology laboratories. Today we're going to mention two different types of H&E staining, but we're going to specifically talk in depth about one. The first one, and primary one that we will be using at TAFE, is a regressive stain. In this method, sections are overstained, as in everything is stained blue, with hematoxylin, then differentiated, some colour taken back out, using acid alcohol. This is to remove any excess hematoxylin from the cytoplasm of the cells, but leaves the colour in the nucleus. Let's have a look. Here's an image of an overstained section. You can see that the dots, the blue nuclei, are nice and dark, but the cytoplasm, the cell background, is still quite blue. After it's been treated with acid alcohol to remove any excess hematoxylin, it looks a little bit like this. You can see now that the nuclei are still nice and blue, but the cell background is clear, or light grey. The other type of hematoxylin and eosin stain is a progressive stain. These sections are stained for a much shorter period of time. They are stained until just the nuclei begin to stain. None of the background stains. Looks just like the second image of a regressive stain. Before we continue to talk about the method, first we must mention labelling of slides. This is to ensure traceability of samples and that the right result goes to the right patient. Don't forget when labelling glass slides to use pencil, and when finally your slides are finished, you can use a sticker and place it onto the label over the top of the pencil writing. All stained slides must have the laboratory identifier or sample number. They must have the patient's family name or surname. They need to have the stain, for example H&E, and the date that the stain was performed. Don't forget to do this, otherwise you'll lose marks. And not to mention you'll misdiagnose the patient. So let's have a look at the procedure in depth. This is a regressive H&E stain. It's the number one stain performed in industry. The very first step we need to do is deparaffinize or de-wax the section. This removes the wax from the sections using HistoClear or xylene. So when we cut our sections on the microtome, they were in wax. So before we can actually stain the slides or the sections with water-based dyes, we have to remove the wax, because if you go back to embedding, you'll remember that water and wax don't mix. So we have to first remove the wax using HistoClear. The next step we need to do is rehydrate the sections or take them to water. We need to remove the HistoClear from the sections using grated alcohols and slowly introduce water back into the sections. So this is kind of the reverse of tissue processing, where we took the water out and replaced it with paraffin wax. Now we're actually removing the paraffin wax and replacing it with water again, so that those water-based dyes can penetrate the tissue. Just remember, HistoClear and water do not mix, so make sure that your reagents are set up in order and that water and HistoClear are never next to each other. So after we've removed the wax from the sections and replaced it with water, we're now ready to stain our sections with some dyes. The first dye that we'll use is Harris hematoxylin. This stains the tissue a red-brown colour. So let's have a look. Have a look at this picture. You'll see that the dots or the nuclei are dark red and the background or cytoplasm is like a light red colour. The following step is to rinse off any excess dye from the slide, followed by the differentiation step. So the slides currently are overstained with hematoxylin. All of the tissue is a red-brown colour. So what we need to do is place the slides in acid alcohol to remove any of the stain from the other tissue components, but leaving the nuclei dark red. So the acid alcohol breaks the dilate complex that is used to stain the tissue, but the strong bonds in the nuclei allow it to remain stained even when we put it in acid. Let's have a look at a slide that has had the excess stain removed. You can see now that the nuclei is still quite dark red, but the background is clear and almost a little bit grey, I guess. We need to make sure that we wash the excess acid alcohol off. Then we can place the sections into Scott's bluing solution. This solution has an alkaline pH. It changes the red-brown hematoxylin into a blue colour. Sometimes it looks a little bit purple. This makes the dye tissue complex, or basically the stain, insoluble. It can't be removed easily except with more acid alcohol. Don't forget to wash in tap water to remove any excess bluing solution. If you don't do this, you'll end up with a white precipitate forming, or basically white dots in your tissue. You need to check under the microscope in the next step. Make sure you check to ensure that the nuclei are dark blue and the cytoplasm should be clear or slightly a little bit grey. You'll notice that this particular slide is still really blue in the cytoplasm or in the background. You can see in here that it's really, really blue. So what we need to do is we need to differentiate again. So place the slides back into acid alcohol and then into Scott's blue and then have a look at them again. Once you have differentiated correctly, you'll notice that your nuclei are dark blue and your cytoplasm is clear or light grey. Now we can move on to the final steps. Eosin is a pink counterstain and is used to stain all of the other tissue components. So the nuclei are already stained with hematoxylin. Now we just need to stain the background a few shades of pink. After staining with eosin, we need to dehydrate and clear the tissues. So this is very similar to what we did in tissue processing. We first remove the water with grated alcohol and then place the sections in HistoClear to remove the excess alcohol. We do this because the mounting media or glue that we'll be using to stick the cover slip on is HistoClear based, which means it doesn't mix with water. Always remember that HistoClear and water do not mix. We can't say it enough. Make sure that HistoClear and water or reagents containing them are not sitting next to each other in your staining set. The final step is to mount or cover slip. These sections need to be mounted with a synthetic resin, in our case DPX, and then a glass cover slip placed on top. Make sure you ask your teacher to explain to you how to get the bubbles out before the following week. If the resin sets, you will not be able to get the bubbles out and the slides will be destroyed over time.